So we're continuing the immune system, lymphatic system, chapter 20 from Amerman. We left off with the beginning of the immune system. Okay, so basically the combined components of the immune system offer three lines of defense against any invading pathogen. So the first line of defense is basically your skin, your cutaneous membranes, and your mucous membranes. They basically act as surface barriers to block any kind of entry of pathogens into the body. So that's your first line of defense. Your second line of defense include all the responses of the cells and proteins that make up our innate immunity. What you see here is a macrophage in the act of destroying a bacteria, a rod-shaped bacterium. That would be part of our second line of defense. And then the third line of defense, which we'll talk about lastly, is the response of the cells and proteins of our adaptive immunity, which are primarily going to consist of your lymphocytes. Your two types of lymphocytes are B cells and T cells, as well as your macrophages and dendritic cells. In figure 2010, you see the pathways for activation of the complement system. The complement proteins are part of that second line of defense. So in this first image, you see complement proteins binding to an antibody that's bound to an antigen. This is the antigen. This is the antibody. This Y-shaped structure is the antibody. And these are the complement proteins that basically activate this process. So that what that does is that causes an enzyme cascade to be activated, and that then activates a complement protein called C3b. Also, another activation of C3b is the lectin pathway, which is a lectin protein that serves to bind certain carbohydrates on the surface of the microbe. And then the alternative pathway is the complement protein C3 spontaneously cleaving C3 to C3a and C3b. And these converge, and all three can activate C3b, which then leads to your activities of your complement proteins. So once that C5b is activated by C3b, then that leads to cell lysis of the microbe. This is a bacteria. The complement proteins are going to assemble and become a membrane attack complex. It looks like that little wagon wheel structure. It then can bore a hole through the cell wall of the bacterium, and that will cause the bacterium then to lyse its contents, ultimately causing the death of the bacterium. Second function is basically the C3A will also, with C5b, will also enhance inflammation by activating your basophils. Remember, your basophils contain histamine granules that can degranulate and release inflammatory mediators, and those will also help. Remember, inflammatory mediators like histamine can dilate your blood vessels, make them more permeable, so that the white blood cells can exit. And then you have C3b leading to neutralization of viruses. These are types of viruses that are coated with this complement protein C3b. That's once it's activated. So these viruses have been neutralized. In other words, they're not lice, but they're not able to produce harm because they're basically isolated from the healthy cells. Another function of complement proteins, it enhances phagocytosis by C3b coating the microbe, this rod-shaped bacterium. And that allows the macrophage easier visibility so that it can then ingest the bacterium that's coated with this complement C3b protein. And then the last function that you see here is basically the clearance of the immune complexes, also through the coating of C3b. And that allows these immune complexes to be more easily eliminated by macrophages. This could be particularly a problem with organs like the kidneys, if those immune complexes aggregated in, let's say, the kidneys, in the kidney tubules. So those are your three pathways that can lead to the activation of the complement protein C3b. Classical pathway, the lectin pathway, then the alternative pathway. This is a close-up showing you the antibodies and the antigens with the complement proteins activating the enzyme cascade, along with the alternative pathway, which involves C3 being activated to C3a and C3b, and then the lectin pathway. All three can activate C3b. That then leads to the complement proteins assembling, producing a membrane attack complex, then boring a hole through the cell wall of the bacterium and then lysing the bacterium. It also enhances inflammation by the complement proteins activating the basophils so that they can then release their inflammatory mediators like histamine, and then makes inflammation proceed by dilating the blood vessels and making the blood vessels more permeable. And then C3b also can coat viruses and neutralize the virus, protecting them from healthy cells. It enhances opsonization, causes opsonization. Opsonization means coating the bacterium with this complement protein, C3b, and that makes the macrophage able to recognize this rod-shaped bacterium so that it can be ingested more easily. And finally, clearance of immune complexes, also by the attachment of C3b protein. So those are all the different activities of your complement protein. So what happens in that second line of defense in inflammation, it basically refers to the cardinal signs of inflammation, like redness or heat, swelling, edema, possible loss of function and pain, and recruitment of the other cells, chemotaxis. Okay. So once that, let's say you've had a tissue damage occurring, let's say a thorn has pierced your epidermis, your skin, and it's reached the tissue area. Well, the damaged cells then will start releasing, along with the mast cells, will start releasing the inflammatory mediators. Mediators like prostaglandins and leukotrienes. These are usually breakdown products of your cell membrane. Mast cells can also release chemical mediators like histamine. And once that occurs, the inflammatory mediators have been released, then they trigger vasodilation of the arteries so that the capillaries will basically dilate and become more permeable to the white blood cells. And this is what results in the swelling and edema that occurs. The redness because of the increased blood flow, and the heat, and then the swelling from the edema from the increased capillary permeability. But those are all secondary to those inflammatory mediators that have been released. So first, vasodilation of the arterioles, then increased capillary permeability. That results in swelling and edema. A lot of edema in this area where the sensory nerve endings, these are free pain nerve endings, and that will irritate these sensory nerve endings and elicit action potentials where you start experiencing pain. Possible loss of function as well. Not always, but sometimes. And then lastly, through chemotaxis, you get recruitment of other cells to the area. The neutrophils will be recruited via the chemotaxis. Those chemical signals that were released during the inflammatory process will recruit more cells to the area. And that'll initiate the second part of the inflammatory response. So once those local macrophages are activated, then the neutrophils then will migrate by chemotaxis, as you see from this illustration, to the damaged tissue. And they'll start phagocytosing the pathogens, the bacteria, the cellular debris, whatever might be in the area. This is an image of a neutrophil undergoing margination, where they travel along the margins of the capillary wall, the arterial wall, and then it squeezes through the endothelial cells, and that process is called a diapedesis. This is an image of a neutrophil squeezing through, and they're basically going to travel to the affected area through chemotaxis. The monocytes also will migrate to the tissues by chemotaxis. This chemical attraction from the chemical mediators that have been released from the damaged tissue. Remember, monocytes, once they leave the vessels, then they can become macrophages. And then they'll start phagocytizing the pathogens and any cellular debris in the area. They'll start cleaning it up. And that also leads to the bone marrow increasing its production of more leukocytes. Remember, the bone marrow is the red marrow, where in the epiphyses areas, the long bones mainly, and that increases the production of more leukocytes, and that leads to leukocytosis, a high white cell count. So that's your inflammatory response, and then your part 2, your phagocytic response. Before we go on any further, I would also like to state that the first and second line of defense, they're basically what we refer to as non-specific lines of defense. They respond according to different pathogens or forms of cellular energy in exactly the same way. Okay, so the types of immunity are classified according to the way it responds to different pathogens or forms of cellular injury. Your innate or non-specific immunity responds to all pathogens or classes of pathogens in the same way. Your innate immune system consists of antimicrobial proteins and certain cells that respond quickly, and it's the dominant response to pathogens for the first 12 hours after exposure. And then the cells of the proteins that exist in the bloodstream, even in the absence of a stimulus. Your components of your adaptive or specific immunity, they respond individually to unique glycoprotein markers called antigens. Antigens are present on all cells in most biological molecules, including our own body cells, and they identify a cell or molecule as belonging to a specific group. Why is your adaptive immunity much slower than your innate immunity that we've just described? Well, your adaptive immunity is slower to respond because one has to first be exposed to a specific antigen for that response to be initiated. And that's how it derived its name, acquired immunity. It takes about 3 to 5 days to mount an immune response. But after that point, it is the dominant response. So there's 2 arms of your adaptive immunity, which we're fixing to discuss here. The first arm is your cell-mediated immunity, which is brought about by 2 types of T cells. The second arm of your adaptive immunity is your antibody-mediated immunity, also called humoral immunity because it comes from the fluids. It's carried out by your B cells and the proteins produced by the B cells, a particular type of plasma cell called antibody. Your adaptive immunity has the capacity, though, for immunological memory, in which exposure to that same antigen is remembered by the specific lymphocytes and antibodies. For example, COVID-19, if you're exposed to that same antigen, and you've been exposed to it this time around, if it comes back around in the fall, then you would have a preferred immunity to that particular antigen if it's the same virus. Many times viruses mutate, though, and when it comes back around, it's in a different form. And that's why it's so difficult to reach a functional vaccine for the H. flu pathogen. So let's look at how T cells mature, and then we'll look at how B cells mature. Remember, T cells are produced in your bone marrow, so your cells of your lymphoid line divide in the bone marrow. The immature T cells then exit the bone marrow, then they migrate to the thymus gland, thymus through its hormones, thymosin and thymopoietin. It stimulates the T cell maturation as well as destruction of self-reactive T cells. Now you've got mature, naive T cells that can now migrate to your lymphoid organs where they take up residence. This is an image showing you how a helper T cell is basically presented the antigen, an exogenous antigen, by, let's say a macrophage that has class 2 MHC receptors on the surface of these antigen-presenting cells, which are usually macrophages or dendritic cells. And this shows you how this exogenous antigen, which originates from outside the cell, is then received by the T cell receptor, the helper T cell receptor. Helper T cells are also referred to as CD4 cells because they have this bridge, this molecule that connects the naive helper T cell with the class 2 MHC molecule. MHC stands for major histocompatibility complex. They're present on all of your white blood cells like your macrophages and dendritic cells. Exogenous antigen means the antigen, which could be a virus or bacterium, means it originated outside the cell. CD8 cells are what we call cytotoxic T cells. They have this molecule called the CD8 molecule that connects its receptor. Okay, these are called class 1 MHC molecules. These molecules are present on all nucleated human body cells, except for, of course, red blood cells. So cytotoxic T cells will involve class 1 MHC molecules from diseased body cells. Okay? And it involves these cytotoxic T cells in recognizing this endogenous antigen, okay, on this diseased body cell. So this is an image, figure A is a class 1 MHC molecule being processed, and it shows you how that class 1 MHC molecule is processed and then displays that antigen onto its surface. Remember, these are endogenous antigens. They're present on all nucleated human body cells. And the second illustration shows you the steps that are involved in processing a class 2 MHC molecule and then displaying an exogenous antigen on its surface. And the reason you're displaying these antigens is so that the white blood cells can recognize that whatever antigen it is foreign and it has to be eliminated. It's kind of like an identity tag, a recognition factor, like when a military wears a certain uniform to basically protect it from friendly fire. Okay. So let's look at first at how the class 1 MHC molecules are processed and then displays their endogenous antigens. So first, the cell synthesizes either a self or a foreign antigen, like you see, because of course it's going to have to start in the nucleus because that's where synthesis, DNA replication occurs. DNA is transcribed to RNA, then translated as the RNA leaves the nucleus, and then it's translated into an antigen. The antigen then is broken down by enzymes in the cytosol. The antigen fragments then are then transported into the rough ER, and then it's coupled with those class 1 MHC molecules in the rough ER membrane. Okay, and then now you've got this MHC antigen complex that now can leave the rough ER by this vesicle, and then it's inserted onto the plasma membrane. Now you've got a displayed self or foreign antigen fragment on this class 1 MHC molecule. So that's how a class 1 MHC molecule will be first processed, and it displays that endogenous antigen. Always remember, class 1 MHC molecules will process and display endogenous antigens. Class 2 will process and display exogenous, which means outside the cell. But these are endogenous, which means they originated inside the disease cell. So this is how a class 2 MHC molecule can process and display an exogenous antigen. Exo means it comes from outside the cell. So this is a viral pathogen. The cell is ingested. The pathogen by phagocytosis. Then the phagocytic vesicle then fuses with a lysosome because the lysosome has the hydrolytic enzymes. The pathogen then is degraded and the antigens are then fragmented. The lysosome then fuses with a vesicle from the rough ER that contains class 2, these are class 2 MHC molecules, and an antigen fragment then binds to this MHC molecule. So now you got this MHC antigen complex that now can be inserted into the cell's plasma membrane. So that's how a class 2 MHC molecule is first processed and then displays that exogenous antigen. And then that leads to, now we're going to look at how T cells are activated, then selected for, that's what we call clonal selection, and then differentiate. Differentiated means to change form or specialize. So first step, your dendritic cells. This is a dendritic cell with its little arms coming out. First, it displays the antigen fragments, okay, on their MHC molecules. Okay, in this case, they're either CD4 molecules or class 1 MHC molecules. Okay, they're either class 2 MHC molecules or class 1 MHC molecules. These are your helper T cells, which are CD4 cells, and these are your CD8 molecules, which are cytotoxic T cells. So first, the dendritic cell displays the antigen fragments on their MHC molecules. This is the MHC2 molecule. And that complex then binds to the receptor of a specific, either a naive helper T cell. This is a naive helper T cell. This is a naive cytotoxic T cell. Naive just means it hasn't been exposed to the antigen yet. Now, in the second image, the helper T or cytotoxic T cell can now bind this other molecule called a co-stimulator, and then it becomes activated. They're on the opposite side of the CD4 molecule, and the co-stimulator on the cytotoxic T cell is on the opposite side of the CD8 molecule. Now you've got an activated helper or cytotoxic T cell clone that can start proliferating, dividing, and then it can differentiate into effector cells, which will actually start destroying the invading pathogen, and memory helper T cells, which can stay around for future exposures to that same antigen. And that's why we develop immunity to antigens that we're exposed to. And that's the reason you have to get vaccinated or exposed to the antigen by natural exposure in order to produce any type of long-term immunological memory. Okay, and this works for your helper T cells as well as your cytotoxic T cells. Those are your two types of T cells, helper T cells and cytotoxic. You have memory cytotoxic T cells as well as effector T cells. Effector cytotoxic T cells will actually be physically involved in destroying and ingesting the invading microbe, whereas the memory cytotoxic T cells will just stay around and be dormant until there's a second exposure to the antigen. So this basically illustrates the effects of your helper T cells. Here's a macrophage with its MHC2 molecules present on the surface. Okay, and this is your activated effector helper T cell. They're called helper T cells because they help both arms of immune response, your B cells and your T cells. Okay, so once that effector helper T cell has been activated by the exposure to the antigen that's presented by the macrophage on its receptor, then that helper T cell can start secreting chemicals called cytokines, okay, such as interleukin-3. Interleukin-3 can stimulate macrophages, which enhances phagocytosis. Interleukin-3 can also trigger macrophages to produce another interleukin called interleukin-12, which then can now stimulate more helper T cells. And that's a good example of positive feedback. Also, interleukin-2 can activate and cause proliferation of more cytotoxic T cells, as well as other various interleukins that can stimulate more B cell proliferation and therefore more antibody production. Antibodies are produced by your B cells. Your activated helper T cell can also directly stimulate your B cell proliferation. And that's why they're called helper T cells. They help both your cell-mediated immunity, which is your cytotoxic T cells, as well as your humoral immunity, which is the secretion of the antibodies by the B cells after they become plasma cells. But they still are very specific for that antigen. This is a close-up showing you how interleukin-3 can be involved in your innate immunity by stimulating macrophages and enhancing phagocytosis. It can trigger macrophages to produce interleukin-12, which then can also stimulate your helper T cells. Interleukin-2 can activate and cause proliferation of more cytotoxic T cells, and they can also directly stimulate B cell proliferation. These are all effects of helper T cells. They can stimulate more B cell proliferation and antibody production, and all of these antibodies are identical, and they'll all be attracted to the same antigen, just like a lock and key. It's very specific. So first, your activated cytotoxic T cell binds to its target cell. This is the receptor. The cytotoxic T cell then releases a chemical called perforin, which then forms pores in the target cell's membrane, plasma membrane. The cytotoxic T cell then releases its enzymes and can now enter the target cell and then start fragmenting the DNA. When the target cell then begins to degrade, the cytotoxic T cell then detaches, and then it'll look for a new target cell. Cells of the lymphoid line will then start dividing in the bone marrow. These will basically cause the B cells to mature in the bone marrow, where the self-reactive B cells are self-destroyed. And mature, naive B cells will then exit the bone marrow and then take up residence in your different lymphoid organs, like your lymph nodes. When they say naive, that means they have not been yet exposed to an antigen, so they haven't been activated. So let's look at the process of first B cell activation, then clonal selection for that particular B cell, and then how it differentiates it and changes form. So first, the B cell binds its antigen and then is activated. Okay, here's your lymph node, here's your B cell receptor, here's your antibodies. Okay, and these are your antigens out here. Here's your class 2 MHC molecules. Okay, so now the sensitized B cell will now process the antigen and then present it on its class 2 MHC molecule. It then binds to a helper T cell, this one over here, which then secretes cytokines that activate this B cell. The B cell then divides repeatedly through mitosis, and then the resulting cells then differentiate into plasma cells, which then can start secreting antibodies, and memory B cells. Memory B cells will stay dormant in the circulation until another exposure occurs. And then once those plasma cells are formed, then they start secreting antibodies that are specific and unique for that antigen that initially activated it in the first place. So that's basically our humoral or antibody-mediated immunity. But remember, for this to occur, first you have to activate the B cells, okay? And that involves exposure to the antigen, okay, that are present on those class 2 MHC molecules. And then once that stimulated B cell processes the antigen, then presents it on its class 2 MHC molecule, like you see in the illustration here. It then binds to helper T cell, which then activates and secretes cytokines that then can activate this B cell. Now the B cell can start dividing and become specialized. It differentiates into plasma cells, which can now produce antibodies for that particular antigen, and memory B cells that'll stay on in the circulation as dormant until they can be reactivated on a second exposure to that same antigen. And then your plasma cells start secreting antibodies very rapidly. Let's look at the antibodies. There's 5 classes of antibodies. There's several mnemonics you can use for these. GAME, G-A-M-E-D, or MADGE, M-A-D-G-E. IgA forms what we'll call a dimer antibody, where they form in pairs. IgA is present in your mucus secretions, breast milk, saliva, tears. Pentamer is when you have 5 antibodies joined end to end like this. So each antibody has 2 antigen binding sites. This green area is the variable region which binds the antigen. So you can have up to 10 antigen binding sites. That's called a pentamer. IgM is a good example of a pentamer. IgM is, by the way, is the prominent antibody involved in your primary immune response. And that's because it's so potent because of its 10 antigen binding sites. Remember, these are the functions of your antibodies. Agglutination, precipitation, where the antibodies clump the antigens together and enhance the phagocytosis of the antigens. Precipitations, you bring the antigens out of solution, they precipitate, and that makes them more easily identified by the phagocytes. Opsonization is where the microbe or antigen is coated. So IgG will coat the antigens on the microbe, and then it then binds to the phagocytes, and that enhances phagocytosis. Neutralization is where the antibodies surround the toxins, the pathological components of the toxins, and then block their toxic effects. Activation of complement is when the antibodies activate complement proteins. These are the complement proteins on the antibodies. That leads to cell lysis by forming this membrane attack complex. And inflammation, stimulation of inflammation. This is where IgE antibody binds to a mast cell and then triggers release of inflammatory mediators. So it stimulates inflammation. So these are just close-ups of agglutination precipitation, where the antibodies clump the antigens together and to enhance phagocytosis. This is opsonization, where the IgG antigens, IgG coats the antigens and then binds the phagocytes, the phagocytic cell, like this macrophage. That enhances phagocytosis. Neutralization, where the antibodies then bind the pathological components of the toxins and block their toxic effects. Complement activation, where the antibodies, this is your antibody, there's your complement proteins. That leads to a membrane attack complex, which then leads to cell lysis. And stimulation of inflammation, where IgE binds to the mast cells and basophils, and then triggers release of inflammatory mediators like histamine. That's called degranulation. So those are your 5 classes: IgG, IgA, IgM, IgE, and IgD. IgG makes up the majority of your antibodies in your serum. It's the only antibody that confers immunity to the fetus because it can cross the placenta from the mother to the fetus. It also functions in opsonization, coating, neutralization, and complement fixation. IgA, in secretions like breast milk and saliva, it mainly functions in agglutination and neutralization. IgM, the first antibody secreted on exposure to an antigen, that's your primary immune response. It's a very potent agglutinating and precipitating agent, and also functions in complement fixation. And then IgE binds to your mast cells and basophils and triggers their degranulation, which thus facilitates inflammation, particularly in the allergic response. And then IgD. IgD is the antibody found exclusively on the surface of B cells, and it also has an important role in sensitization of B cells and then subsequent activation. Here's IgG, makes up the majority of the antibodies. IgA, which is a dimer. IgM, IgE, and IgD. Okay, let's look at a comparison of the primary and secondary immune responses. Your primary immune response is when you're first exposed to an antigen, like a virus, like you've been exposed to COVID-19. Notice the IgM is your prominent antibody. Okay, it has a lag phase of about 4 to 5 days, and the time until complete antibody peaks is for IgM is about 1 week, and for IgG it's about 2 weeks. And the duration of response is between 14 and 21 days. The secondary immune response on the right here, notice the lag phase, that's the lag phase is the time you don't have symptoms. It's only 1 to 3 days, okay? So it's much more efficient, okay? The time until antibody peaks is basically 3 to 5 days, as you can see. IgG is the primary antibody, as you can see, and the amount is much higher than what you see in the primary immune response. And its duration is from 28 days and beyond. So it's got a much more thorough response. So antibody-mediated immunity can be divided into active immunity and passive immunity. Active immunity is when you make the antibodies to the antigen. It can be either naturally acquired through exposure, just from an infection, where you're exposed to somebody that has the antigen or the virus, or it can be artificially acquired through vaccination, like when you get a vaccination for H flu. In this case, you always have memory cell formation, so therefore you have lasting immunity, lasting protection. Passive immunity is when you basically receive antibodies from another individual that's already produced the antibodies. It can be either naturally acquired through passage from mother to the fetus and in breast milk, or it can be artificially acquired through injection of antibodies to toxins or venoms like a snake venom, antivenom. And of course, the result, there are no memory cells formed, so there's no lasting immunity, no lasting protection. Okay, then the last figures, we'll talk about the immune response to the common cold. First, the infected cells, like from a virus, will trigger an inflammatory response, and that secretes the interferons. Interferons then prevent the neighboring cells from, and they activate the natural killer cells, okay, which then lyse the infected cells. So they prevent infection in the neighboring cells, but they also activate the natural killer cells. Then the dendritic cells then activate the helper T cells in your lymphatic tissues and your cytotoxic T cells. And they also activate your B cells that are bound to your viral antigens. So the B cells will start differentiating into plasma cells and start producing their antibodies, which then start neutralizing and destroying the viruses, okay, which you can see here. The naive helper T cells also activate your helper T cells to secrete cytokines, which stimulate the cytotoxic T cells to basically amplify the immune response, and the cytotoxic T cells then start destroying those virally infected cells, as you can see from this image. So that's how your immune response occurs in, let's say, a common cold exposure, which are, of course, viruses. Now let's look at the immune response to a bacterial infection. First, the bacteria will damage the cells, and the inflammatory response is induced. Inflammatory mediators are released, and the local macrophages start ingesting the bacteria. Neutrophils then are going to enter the area in large numbers. That's where you get the high white cell count. In lymphatic tissue, the dendritic cells then activate the helper T cells, and the B cells will bind to the bacterial antigens and are then activated by your helper T cells. Okay. And then the B cells then differentiate into plasma cells and start secreting antibodies that start agglutinating the bacteria, whereas the helper T cells start secreting cytokines. These are the cytokines that enhance the phagocyte as well as the B cell activity. And then cancer cells. Cancer basically is going to damage the surrounding cells and induce an inflammatory response. Okay? Natural killer cells in the area will then migrate to the area and then begin to destroy the cancer cells. Dendritic cells will start ingesting the cellular debris and migrate to the lymph nodes, where they then activate the naive helper T cells and the cytotoxic T cells. The activated helper T cells then secrete cytokines that then stimulate the effector T cells to start killing the cancer cells, as you can see from the illustration. So in hypersensitivity, type 1 hypersensitivity, the first exposure is when the antigen, the allergen, first binds to a B cell. The B cell then differentiates into a plasma cell, which then starts secreting IgE antibodies, which then bind to a mast cell, and what they do is then degranulate the sensitized mast cell. The allergen binds to the sensitized mast cell. This is the allergen. This could be pet dander, dust, mites, pollen, whatever. And IgE molecules on the cell then form crosslinks that then cause the cell to release inflammatory mediators like histamine and serotonin and bradykinin, and that triggers the inflammatory response. This is the skin of an AIDS patient with Kaposi sarcoma. AIDS is caused by the human immunodeficiency virus, and it was actually AIDS was incidentally discovered initially by noting Kaposi sarcoma on young males that normally would not come down with a Kaposi sarcoma. Kaposi sarcoma only generally affects, before the AIDS era, only generally affected elderly individuals, usually elderly men that had a weakened immune system. But when they first saw this Kaposi sarcoma in young males, they knew that something was definitely not right with the immune system. So, and that led to the discovery of the human immunodeficiency virus in Paris.